Okay, so welcome everybody. Welcome to this research webinar organized by the Department of Rehabilitation Science here at the Polytechnic University in Hong Kong. My name is Georg Kranz and I will be the moderator today. And the topic of today's webinar is brain mapping using magnetic resonance imaging. And this is presented by Dr. Edward Hui, if I'm correct, Hui. He's assistant professor at our Department of Rehabilitation Science, and Dr. Hui specializes in MRI. He has published many papers on MRI in also very high-ranked journals, and I think he focused on diffusion-weighted imaging, and I'm sure he will talk about that in a moment. So today he will give an overview on brain mapping and on the latest developments in MR imaging. And he will talk about 45 minutes, he just told me, and then we will have 15 to 30 minutes Q&A. Okay, and now I will hand over to you, Edward. Please go ahead.
Okay, thank you. Thank you, Georg, for the introduction, and good afternoon, everyone. First of all, I'd like to thank the Department of Rehabilitation Sciences for giving me this opportunity to give this seminar. And just as importantly, I'd like to thank you all for taking the time to attend this seminar, especially we have a diverse group of our audience, which is really wonderful and motivating. So as a disclaimer, I was actually trained as a biomedical engineer with special interest in developing imaging biomarkers and methods for neurological applications, both in terms of preclinical and clinical application, actually. And in this seminar, I will first give you some background information and the latest development of MRI for brain mapping. And the primary goal of this talk is really to convince you that MRI is the right tool for doing research in the context of basic and applied neuroscience, as well as understanding disease mechanism. Because of the time limit, some use cases of MRI will be briefly mentioned, and I'll elaborate a little bit more on those that are of emerging importance. And overall, I will not give too much technical details, but I'm happy to explain more if you have any question during the Q&A session. So, so this is the outline of today's seminar, and let's dive right into the MR basics. So I'd like to first draw your attention to the limitation of MRI in terms of the spatial scale. MRI can only allow us to look at biological tissue on the order of 100 micron up to meters. But technically, we could actually go to higher resolution in around this region, but at the expense of experimental time. In other words, if you want to achieve much higher resolution than is typically performed, you will need to scan the subject for a much longer period of time, which is obviously not very ideal. The key advantages of MRI, or pros and cons of MR, are that the pros of MR is that the subject are not exposed to any radiation, so it's very safe, and it offers exquisite soft tissue contrast that no other imaging modality can easily match. Notice that soft tissue contrast mean it can be from both endogenous contrast or exogenous contrast, meaning that we can get exquisite tissue contrast even without injection of chemicals that can enhance your body. On the flip side is, as I alluded earlier, MRI is actually very slow. A typical MR exam takes around half an hour, but other techniques such as computed tomography, which is one type of X-ray technology, may take minutes of your whole brain. So it's very slow, and it's susceptible to artifacts and very high cost. So MRI can be applied to every organ of your body, basically, and all the internal organs except your lung, as well as your extremity. So basically everything except your lung can be imaged. Because of MR's versatility, it's really an ideal tool for clinical practice in terms of performing routine clinical diagnosis, as well as clinical treatment monitoring. And it's also an ideal tool for performing basic and applied neurosciences research or other type of applied or basic researchers. And I will provide some example throughout this seminar. So our university has recently installed a new 3 Tesla MR scanner, which is, I believe, one of the 2 most advanced systems in Hong Kong, as shown in this picture. This is the real picture of our scanner. And our scanner has full research capability. And as you may notice or have heard, MR scanners that have different Tesla, like 1.5 Tesla, 3 Tesla, 7 Tesla. This is actually denoting the field strength or the strength of the magnet. Basically, very loosely speaking, you can understand it as the scanner's field strength is actually related to the image quality, to the signal quality that you get as shown in this diagram. So loosely speaking, the higher the magnetic field, the better the image quality. And as I mentioned before, the scanner that we have is a 3 Tesla scanner, which is around in this region of image quality. And there are actually some other research center in the world that has 7 Tesla machine, which has roughly 50% better image quality, actually double the image quality. But even though we are using 3 Tesla scanner, actually with the advantage of MR technology, the image quality that we can get nowadays has been tremendously improved, especially for the brain. So I'll demonstrate to you some examples again. So in essence, we don't really need 7 Tesla because it's really expensive. It's high maintenance and it requires a lot of expertise. So this diagram shows you a very nice, vivid example of what I mean by using different type of scanners across time. So what is shown on the top row of images is a very popular imaging biomarker of the brain, which is called fractional anisotropy, or in short, FA. What it shows is the white matter tracks. Anything that is bright can be considered as white matter tissue, and anything that is dark is likely gray matter. And on the bottom, it's kind of similar information, but it shows you the color of the white matter track. The color actually denotes the direction of the white matter track. So the red indicates the white matter track going from left and right. Blue indicates the white matter track going from superior to inferior or the other way around. And green is anterior posterior. So on the left, you can see it was acquired from one of my stroke patients at 3 Tesla, but the scanner was pretty old actually. So the resolution that we can get was pretty abysmal, 3 millimeter. But for relatively current clinical scanners, you can perhaps, or more advanced clinical scanner, you can get up to 2 millimeter. But with the latest 3 Tesla technology, which is what we have currently at PolyU, you can actually get much better resolution, 1.25 millimeter. I mean, without any training, you can already tell the difference in information is dramatic. Like in here, you don't see very well differentiation between some of the white matter tracks, but in this case, you see really clearly. So in essence, it tells you the MR technology has come a long way in the past 2 decades. So let's move on to the topic of this seminar, which is what we can really map using MRI. As I mentioned to you before, in terms of the spatial scale, which is the limitation of MRI, it can be made very sensitive to brain microstructure on the order of 100 micron, all the way up to the entire brain, which is kind of like microscopic. And over the past few decades, there are numbers of brain imaging biomarkers that have been developed, and I will mention some of them in the subsequent slides. So this is illustration of the brain microstructure and the overall brain structure on the macroscopic scale. But I just want to point out to you that when I say we can measure microstructure, we are actually indirectly measuring the microstructure. And whereas the macrostructure, obviously, we can measure it kind of relatively more precisely. So let's start by talking about the imaging biomarker of the brain that gives you information on the local brain structures. First up is we can actually use ML to obtain volumetric information of our brain. Because of what I alluded earlier, again, in terms of the development of ML technology, we can now achieve images that are of very high resolution. In this case, this is on the order of 0.75 millimeter. So at this resolution, it's very easy to discern the boundary between gray matter and white matter, which would then allow us to quantify most obviously the volume of different tissue, especially in this case, we can then quantify the cortical thickness of the entire brain. And in fact, this cortical thickness has been shown to be a very useful biomarker for a number of neurological diseases and disorders such as Alzheimer's disease and schizophrenia. Right, so as I said earlier, we can actually measure the cortical thickness of the entire cerebral cortex, and the color indicates to you the warmer the color, the thicker the cortex, and the colder the color, the thinner the cortex. And as you can see this on this histogram of this cortical thickness distribution, you can see that the distribution is somewhat normally distributed. And I would like to draw your attention to the fact that the mean thickness of our cortex is about 2.6 millimeter. And remember I said about the ML technique, ML development. So in the old days, we typically study the brain activation or the activity of our brain using a technique called functional MRI. The resolution that we acquire typically is about 4 millimeter. And based on this histogram, you can tell with 4 millimeter, the maximum that you can get in terms of the number of pixels that span across your cortex is only 1 pixel. And you can imagine the reliability that you can get with that sort of resolution. But with the latest technology, we can actually go up to 2 millimeter. And the amount of information will be subsequently much higher, and I mean in terms of the specificity. So of course, if you are more adventurous, you can always use even higher or better scanner at 7 Tesla. You can get up to maybe 1.6 millimeter resolution. So another type of imaging biomarker of the brain is related to myelin content. We can actually use MRI to measure the myelin content of the cerebral cortex, and I'll give one implication of this myelin mapping in another slide later on. So remember I mentioned in terms of clinical application, MRI is really good for looking into treatment monitoring. So one of our earlier study was to apply the fractional and anisotropy map that I mentioned to you before, which is a map of white matter, to study the treatment efficacy of an experimental drug for the disease called multiple sclerosis. So it was, as I mentioned before, FA is a nice way of visualizing white matter, and to turn it around, it means that we can use it to monitor white matter changes due to a pathology, such as in this case, demyelination, which is in the dorsal column of the white matter, spinal cord of a rat. And we can then monitor the treatment efficacy of this particular experimental drug, and to indicate to the readers that, well, the white matter was relatively intact. So that means possibly this new experimental drug was effective in stopping or at least reducing demyelination. So another clinical application of MRI is to look into stroke assessment. In fact, it was one of the early clinically proven technique for stroke diagnosis. And what we did in this earlier study was to propose a new biomarker, which is called mean kurtosis. In this case, we don't have to know the details, but what we did was we proposed a new imaging biomarker to study the evolution of infarction in animal model of stroke, which was really nicely depict some contrast that you don't usually see from conventional techniques. And we later on translate this new biomarker to study stroke patients, which was really exciting. Not only can we use MR for disease diagnosis, but we can also use it to do some research, such as to look into the pathophysiological changes due to certain diseases. In this case, we apply the same new biomarker that we proposed for short application to study a relatively new disease called relatively new pathology called microinfarction, to study the evolution of the infarction, and we see something very dramatically different from what was from conventional techniques, basically. Because of the time, I can't cover all the basic local brain mapping techniques, but these are some of the lists that I can come up with, and they in fact the type of contrast that we can get is wide ranging, going all the way from, like, we can measure the brain perfusion, or also measure the amide content or macromolecule content, or iron content, and so on and so forth. So in a way, MRI is, again, a very, very robust technique and versatile technique that allows you to measure different information of the brain, and actually other organs as well. But majority of the investigation that I showed you previously focus on local brain changes. And but what about if we can come up with a way to study the relationship between different brain regions? And with that, I will simply call these biomarkers that map the brain on a global scale to refer to the biomarkers that allow us to study this information, study this relationship between different brain regions. And the primary reason why we wanted to look at the relationship between different parts of the brain is actually very nicely summarized by this New England Journal paper. Essentially, it was believed that the relationship between brain lesion and symptoms was not as straightforward as previously believed. What I mean by that was that, on one hand, there were a lot of patients that presented with very similar symptoms, and they were found to have very similar lesions that are located in, they were found to have lesions that were located in similar region of the brain. But subsequently, there were other cases where the patient who have similar symptoms, but then the lesion that was found was actually located in very different part of the brain. In this case, shows you like somewhere in the frontal cortex, somewhere in the parietal cortex, and so on and so forth. So this is the reason why we wanted to look at or develop techniques to study different regions, the relationship between different regions. And so how do we do that? We can actually study this information in terms of the anatomical information or anatomical connection between different parts of the brain, i.e. the wire matter connection across the entire brain. So these lines that I showed, so these lines are kind of similar to what I showed you before, the color FA map, where the color indicates the direction of the wire matter tracks. So these lines are all wire matter tracks, or apparent wire matter tracks. On the other hand, we can also measure the activation of the brain activities, or in terms of the connection between different parts of the brain in the context of their brain activations. So this is what we call functional connectivity, and the previous case was structural connectivity. In fact, we have two ways of measuring the functional connectivity. One is we can measure the brain activation while the subject is at rest, meaning they don't need to do anything but just sleep in the scanner. Or we can ask the subject to perform some tasks, and we can then look at their brain response to these tasks. And these are two major types of brain connections that we can study. So one is anatomical information, one is more or less physiological. So now that we have this nice way of globally characterizing the brain or the brain connectivity, how do we actually use a very systematic way to characterize these information? And Sporns et al. Have proposed coined this term of Connectome, which he borrows the term from omics, where this is essentially a repertoire of all the connection in your brain. So how do we actually get this connectome? And on the left, you see this is a schematic diagram of your brain, and different different parts of the brain have been kind of like segmented or separated. So these different boundaries, kind of like the world map where different countries have different boundary. And in this case, this is different regions have different boundary, and they are indicated by different color. So what you can then do is very trivially, you can first study the connection between one region, which is what we call the seed region, the green region here, and then the target region, which is kind of like the pinkish target. And then you calculate the number of connection between the two, which in this case is schematically 4. I mean, as I alluded earlier, we can count the number of anatomical connections, or look at the number of, or the strength of the functional connections. And then we can repeat this process and to look at other brain regions, say in this case, again, the same seed, but then different regions, the brown regions, and then we get 10, and then the blue region. And then we repeat this process by exhaustively go through all the regions and to compute this matrix, which is Connectome matrix. And in a way, this is a classical, typical Connectome that you can measure. And in this case, this is a structural Connectome, that is the connections, the anatomical connection of the entire brain. This was collected from one of my stroke patients. So now that we have this Connectome, which is a very systematic way of storing all the information regarding your brain connection into one place, then how do you do subsequent analysis? So one way to do it is you can then represent the brain in terms of a very classical ball and stick model, where the ball represents the different brain regions and the stick represents the strength of the connection between different parts of the brain. And actually this kind of diagram, or this kind of model, is very well known as a graph or as a network. So I'll interchangeably call the brain as a brain network or brain graph. So they mean the same thing in this context. So now that you can characterize the brain as a graph or as a network, what you can then do is kind of like apply in different contexts. So in this case, we have recently published a study where we use this brain network to study the brain network. Study the effect of brain stroke recovery on the relation between this brain network and stroke recovery. And we found that the organization of the brain actually changed quite a lot across the course of stroke recovery. And another example where we also can use this information in terms of the brain network to kind of come up with some biomarker of the brain network that allow us to potentially predict the outcome of the stroke patients. This is again under review, and this is the hope of predicting stroke recovery using brain connectivity, basically. And as I alluded earlier, apart from looking into performing some clinical diagnosis or some prognosis, we can also use MRI to investigate some new diseases or relatively new diseases. In this case, we want to look at the effect of a relatively new neurological disease called small vessel disease, or look at this effect of SVD on the brain structures, essentially. So I think I'm going pretty fast, but it's okay. Remember, regarding functional connections, we have two ways of measuring the functional connection. One is to measure the connections while the subject is at rest, or in other words, it's called resting state connections. And the other type of connection is task-induced connections. But what happens when the subjects cannot perform the task that you really want? Say in the case of, for example, patients that are not able, say for patient with acute stroke, they might have certain neurological sequelae that prevent them from performing, say, motor tasks. Then you can't really perform task-based studies. What you can then do is just resting state connectivity, study their resting state. And this study by Taver, published in Science, shows a very nice example of actually what is the value of resting state connections. He showed actually you can use the resting state connection to predict the response of individual subjects to certain well-known tasks. So in this case, this is showing the prediction of the a social task from simply simply collecting their resting state connectivity, and it shows you quite nice correspondence between the prediction and the actual task performance. And in fact, this kind of prediction was shown to be really specific to individual subjects. In this case, this stuff in the same given task, language task, for example, subject A doesn't have any activation in this region of the brain, but then in subject B, it has activation. And this kind of model prediction allows you to predict this sort of task variability. And in a sense, it suggests that the resting state connectivity contains some really valuable information of your brain. And again, we're talking about resting state connection. One of the study was to sort of understand whether there is any systematic difference between the resting state connection of different individuals. And what this study did was to repeat the same data collection across different sessions. So this is one subject, this is another subject, and the different roles indicates the measurement of the same subject at different time. So you can see the correlation between different time is actually pretty strong per subject. So the warmer the color, the stronger the association between different resting state brain connections in different experimental session. But if you correlate the resting state connection of one subject to another, the correlation is dramatically different. So this indicates to you actually perhaps the resting state connection contains some individual specific information that may allow you to do certain things that I'll try to allude to. I mean, try to put forth in the outlook towards the end of this talk. So remember I mentioned about measuring the myelin content of the cerebral cortex, and this same study actually found out he sort of alluded to the variation in terms of the resting state network of different subjects being also underpinned by the variation in the myelin map. I mean, the myelin content of different cortical area. So these color maps indicates the myelin content, and the boundary indicates the resting state network. So he kind of suggests that the variation on these sort of boundary or the resting state connections is actually maybe attributed to the variation in the myelin concentration across different subjects. So this is really interesting, and to suggest to you that MR is really powerful, and the information content that we can get from MRI is potentially very subject specific and maybe physiological in some cases. And so remember, we have two types of network, right? We have structural brain network. We have also functional brain network. But then they were often measured differently, or they were not studied as the relationship between the two networks wasn't really studied very extensively. But then in this particular study, they proposed a way to kind of propose a biomarker to look at the relationship between these two networks. They call it coupling, meaning they simply correlate the structural connectome with the functional connectome and try to find this marker of coupling to see if it's specific to any particular physiological change. In this case, they were trying to find out whether there's any association between this coupling and age. And in fact, there are some striking correlation and it's some positive correlation, some negative correlation. But more importantly, they found this age-related association with coupling has some implication in terms of which particular function of the brain is most that underpins this relationship. And in fact, they found one type of network called default mode network, which is a relatively well-known network that is responsible for higher cognitive function of our behavior. And they found this particular network has a very statistical significant correlation with the this age-related association. And this is kind of interesting. So they look at the coupling and to see whether the coupling has some implication in terms of whether it can explain the variation in the performance of executive tasks of certain of these subjects. And in fact, it was well known that the executive function of young adults is related to their age. And with this study, they found actually the coupling between structural and functional brain network has some mediation effect on this age-related executive function, so which is rather interesting. I guess I'll move on to the kind of towards the last part of the talk, which is I kind of propose to you, or at least based on what I understand from the literature, the investigation that are worthwhile in the next 5 years. So remember, now that we can perform the brain mapping with a much higher resolution, actually this is great. This is not the same as not analogous to the case of photography, where some people argue the image resolution that you get from a picture, the higher the resolution of your scenery doesn't necessarily mean that the quality or the information content that you get is higher. But in the case of MRI, I would argue it's quite not the same, and it's actually we really need high resolution to obtain certain information. Say, for example, in this case, you can, for those who have very careful eyes, if you focus on this region of the brain where actually the white matter tracks are actually traversing in different directions, meaning in other words the architecture of the white matter is pretty complicated in this case. But with high resolution, we can sort of differentiate different white matter population. But if we were to study the brain at this very coarse resolution, we only can get some much different information. And in fact, this information could be possibly wrong. So in essence, with this new or better scanner, we can have a lot of information. And as I alluded in the previous example, where now that we have this wonderful way of studying our brain in terms of studying their anatomical connections or functional connections, can we actually build a different brain model, or in terms of as in to understand, improve our understanding of the pathophysiology of the disease, say, or to improve our disease diagnosis? And in fact, in the past like 5 years, there are a substantial number of studies to do just that. So they hope they can use the brain connections to allow them to predict or differentiate patient with say who may progress to AD or from those who will not. And also it has been a challenging research topic to kind of infer behavior from our brain. Essentially, perhaps maybe we can one day use this sort of extensive information regarding our brain to understand this structure-function relationship. And in fact, one of our ongoing study is to hope that we can use one type of deep learning, which is relatively new, and I'm happy to discuss in an offline setting, is to sort of, given these sort of brain structures or brain connections or brain networks, can we predict the outcome of these, say for example, stroke patient? If we measure their brain at the acute setting, perhaps can we use deep learning to first of all allow us to predict which patient everyone has the same postulation, meaning we apply the same postulation to all the individuals. But actually this problem, it was potentially wrong, because from this study, it tells you that the segmentation of different regions is actually different across different subjects. And he also shows you that perhaps this differences in brain postulation may allow us to explain the differences in task performance or task response of different subjects. So this is really interesting. And I guess with the interest of time, I'll stop here, but I'd like to acknowledge a lot of people, because without them, I would not be able to do some of the research that I show you, especially my mentor, Professor Chen, who was the one along with the department to recruit me to this to University of Polytechnic University. So and also like to acknowledge some funding agency from NIH from the US and Hong Kong funding agency. And I'll take this opportunity to sort of do an advertisement where we have an opportunity for the young or young and upcoming scientists to join us to study different problems, because I don't just look at brain connections, I also do some other stuff. So you're welcome to contact me if you are interested. And lastly, I'd like to mention, for those of you who would like to explore into MRI and to do something differently in terms of understanding our brain, you're welcome to collaborate with us, because as you can see in this diagram, I try to indicate to you that we have quite diverse experiences with different type of scientists, like from clinicians to radiologists to engineers, and also psychologists. So we are happy to collaborate with anyone who with diverse background in the realm of brain imaging. So with that, I'd like to stop here, and I thank you for your attention, and I'm happy to answer any